Yeah, I want to talk to you a little bit about what we've been doing over the past year. And so Angela emailed me about a month ago and asked if I could come up and tell you what I've done, give you a short description of what the science team has been up to since our last DNAxis Connect meeting. And I got a little bit scared because I don't do short very well, and B, we've done a whole lot, so I was concerned about how we're going to fit it all in. So I was thinking about, should we tell stories about some of the portal work we've been doing, some of the challenges we put together for Precision FDA and our microbiome platform, Mosaic? Should I talk a little bit about the St. Jude Cloud? Talk some about the work we've done with partners like PacBio. They've come out with some really excellent upgrades to their tool set in the past year. And talking to Svetlana earlier this morning, it sounds like they have even more to come this year. Talk about some of the engagements we've gone into in Synteon and benchmarking their variant calling tools. We've had a great year with our cloud partners. Had some really awesome projects in the clinical space with Melbourne Genomics Health Alliance getting their somatic and germline pipelines put together on the platform. Work at Baylor and UPMC and Regeneron. We had a launch of a new clinical trial solution. Like we talk about, we've started supporting CWL and WDL in our workflow descriptions, and additional, it get on and on and on. But then I saw the agenda, and I saw most of these items were already taken by other speakers who can talk much more eloquently than I can. So that was good. So I decided to talk to you today about four main topics: benchmarking, some assembly work that we've done over the past year, some visualization stories, and then wrap it up with a new tool that we co-developed with our friends at Baylor called Parliament 2. So first, talk to you a little bit about benchmarking. So benchmarking, I mean this in sort of the broadest sense. It's kind of what I think most of the bioinformaticians in the audience probably like to do on a daily basis, is take out new tools, kick the tires, take them around the corner, see how they perform and how they behave. So at DNAxis, we get the opportunity to kick a lot of tires. We play with a lot of different tools, and we are engaged in a lot of different projects. So over the past year, just a quick shout out to our blog. We try to capture a lot of this work on our blogs and tell you what we've observed and what tools we find work really well in some of the work that we're doing today. We've had blogs this past year talking about JDK4 when it was first released, talking about this new compression technology that will compress your BAM and CRAM even further, looking at new instruments that have been released over the last year, talking about deep variant. But the thing I want to spend a little bit of time talking today about is evaluating the performance of next-generation sequencing pipelines. So over the past few years, I think the community as a whole has done a really great job of developing new standards that allow us to evaluate how well we're doing in the field of sequencing. In particular, consortia like Genome in a Bottle have created these great truth sets that we can use to evaluate both our sequencing instruments, sequencing processes, and then variant calling pipelines. So we were talking in our weekly science meeting one week, and my colleague Andrew pointed out that he had some concerns about some of what we were doing in this effort. He said, this is great, we've made a lot of progress, but when we are presented with the raw data to evaluate the variant calling pipelines today, we're kind of presented with the best possible data you could ever see. So in my mind, this is kind of like if you wanted to evaluate how good my handwriting is, it would be a mistake for you to look at the envelope that contained the wedding invitation that we sent out, right? That had this really elaborate calligraphy, and as anybody who has had to try to decipher the chicken scratch of my notes knows, that is not in any way relevant to evaluating how good I'm able to do handwriting. So in the same way, if you look at the FASTQ files that are made available for some of the samples in Genome in a Bottle truth sets, what you find is that they are uncharacteristically really good samples. They knew they were being tested. They studied for the test. They did all the preparation. And then they gave you a really great result. But that's not exactly the world we always live in. So what you see here in this chart is a distribution of the expected number of errors one would find in 100 base pair reads from the raw data made available for a lot of the genome in a bottle data sets. But then if you compare it instead to, on this chart, look at the blue samples clustered at the bottom there. Those are all the genome in a bottle data sets. Error rates, less than 1 base pair per 100 base pairs sequenced. But then the red dots reflect the expected number of errors you would see in just random data we extracted from the SRA, right? So significantly higher, much different distribution. And so the question is that if we're evaluating our processes, our variant calling pipelines, based upon this abnormally perfect data, are we missing something possibly? So we developed this tool called Readshift to help us try to answer that question. So what Readshift does is it can take actual real-life sequence data from truth sets like genome in a bottle and try to back out the goodness of them, right? So if you have, in this case, the blue curve there shows you can think of it as the quality scores distribution found in a 300x coverage worth of data from the genome in bottle data set. Then when you downsample that data to pass it through your variant calling pipeline, you can preferentially select some of the poorer reads, or the reads that are more akin to what you would find in the real world. And so what Readshift does is allows you to sort of have a dial there, and it allows you to dial in how many standard deviations away from the ideal good distribution there in the blue do you want to move over to the not as good data. And then once you have that data set, again, what I want to stress here is that we're not actually changing the data itself. The reads are coming out. These are true reads coming off the instrument. We're just preferentially choosing some of the ones that had more errors in them as reported by the instrument itself. So when we do this, what we can do then is pass that data through the various pipelines and see what effect the various worsening of the sequencing data has on the different pipelines. And what you see is it has a dramatic effect. So in this chart here, this laser is not bright enough, but in this chart here you'll see 6 different variant calling pipelines that we evaluated with different quality distributions of data. So JDK3, JDK4, Cintiyon, Deep Variant, Strelka, and Freebase. In blue and in red, you see basically unperturbed data. So this is the pristine, good genome in a bottle data. But then as you move across the color spectrum, you see us preferentially going worse and worse data out of there. And as you would expect, your ability to accurately call variants gets worse the worse data comes out. But it's not uniformly worse across various different pipelines. And so you can see that in particular, the one that I found most interesting here was tools like Strelka, where at really pristine data, it was one of the better variant calling pipelines. But then as the data gets to be more realistic and what you would really see in the real world, you get worse and worse and into some really crazy different results as you get farther and farther away. So I thought that was kind of interesting. What is also interesting, that was for SNP errors. But then for indel errors, we don't observe the same sort of pattern there. So in the indel errors, confusingly, for some of the worst data, you actually get a little bit better than you would for the pristine data. I'm not exactly sure why that is. Our best guess internally on why we think indels are less susceptible to the worsening effects that you see with the SNPs is because oftentimes the errors in these instruments are errors at a single nucleotide level, and if you're looking at indels, that's an event that incorporates multiple loci, and so you're able to sort of correct for that or something, right? The other aspect that was kind of interesting on this is that not only is the accuracy of the variant calling pipelines impacted by the quality of data that's presented to it, but also the runtime to actually get your answers to begin with, or potentially it could be impacted. So this is kind of an interesting one as well. In blue, you see data that came off of a HiSeq 2500 instrument. And then in red, you see data that came off of a NovaSeq instrument. You can see here that for the HiSeq data, the quality of data that you're examining doesn't seem to have much of an impact on the total runtime. But on the NovaSeq data, as you get the data migrates away from the pristine data, the runtimes can increase fairly dramatically. The best guess we have internally for why that is, as many of you all are probably familiar, the NovaSeq instrument uses a new chemistry, a 2-color chemistry, to call bases instead of a 4-color chemistry like some of the older HiSeq instruments from Illumina. What impact this has on it is that the instrument itself is unable to differentiate a no call at a given position from a, I forget what letter it is, but let's call it C, at that point. So you probably preferentially see more errors of that particular base pair, and that may result in those reads piling up at say a CG-rich region in the genome. And then the variant calling pipeline, in this case GADK, might have a large stack of reads that it has to go through and call variants on. Anyway, I thought that was kind of interesting as well. Okay, so that was a quick story about the benchmarking efforts that we've been up to over the past year. The next story I want to transition over into is genome assembly. So we've been very lucky over the past 2 years or so to be able to be involved in a lot of de novo genome assembly projects with various partners of ours. So over the last year, we've assembled a lot of different species from plants to amphibians and reptiles, like you get your frog there, various insects like the boll weevil, cow, and various other organisms. In fact, over the past year, we've done over 50 de novo genome assemblies. That's over assembly per week on average. And for those of you who may not be familiar with the assembly process, I find it very, very interesting from a bioinformatics standpoint. And there's a number of reasons for that, but one of them is for what all is involved in it, right? So the typical variant calling pipeline is akin to having a puzzle. You have a whole bunch of reads, or these puzzle pieces, and you want to put it together and figure out, what makes me, what makes Omar, and what have you. But we have a pretty good idea what the picture is going to look like at the end, right? We've got a reference genome to go by, so we can look at the puzzle piece in hand, say whereabout does this belong, and solve our puzzle that way. Well, for de novo genome assembly, you got the puzzle pieces, but you're missing the picture, right? So you don't have the ability to know this read, where am I going to put at the end? So it becomes this problem where you're taking every single puzzle piece out of the box and trying to figure out where they go just by holding them up to each other. Does this fit? Does this fit? Does this fit? Computationally, you can imagine that that's a very complex and challenging problem to do, which is great for the cloud. So that's why we really get excited about it in the science team. Because we can spin up thousands of computers, as we were talking about earlier, and have them all going concurrently and get the problem solved in a pretty quick time frame. Also, we like to imagine ourselves as mini Francis Collins, and we get to do a de novo genome assembly in a week, whereas it took them multiple years back in the early 2000s, right? So it's an exciting project to get to do. So we've been involved in a number of different projects this year. This one is kind of an exciting project that we got involved in because we attended the PacBio Smart Leiden event back in the spring. So there, we asked folks there to nominate their own project that they were working on and explain why they thought their project should get a free assembly on our cloud platform. And so this project that won was from Charlotte Bartley Cross, who's at the University of Sheffield in the UK. And she's working on this Seychelles warbler project. So I don't know a whole lot about this bird, but apparently this bird lives on, I think, some of the islands in the Azores, or the Seychelles rather, islands in the Seychelles, and it had gotten down to only a couple dozen of individuals left. But this Seychelles warbler consortia came together and started doing some protection work on it. And they've now grown it up to 5,000 or more individuals of the species, and they've introduced it into multiple islands across the Seychelles. And so we were really taken with the overall story. We were able to take in her data and get an assembly put in place for it. So now she's going to be able to use that reference genome assembly to monitor genetic diversity across this group of warblers that's gone through this incredible bottleneck, right? And so now they're able to use that data to make sure that their sort of expansion project is going to work out well, and will hopefully be able to have the Seychelles warblers for generations to come. Another project that we got to do actually around Christmas time of last year was a de novo genome assembly of wheat. So those of you may not know, wheat is a very challenging organism to work with. It's 16 gigabases in size, and it's a hexaploid species instead of diploid. So it's about 5 times bigger than human, but as you can imagine, computationally, it's not just 5 times more complex. Because you have to take every piece out and compare it to every other piece of your puzzle when doing the assembly, it's actually almost 25 times more challenging to assemble, which, again, was really exciting for us. So we end up over 2 weeks consuming over 1 million core hours to try to put this puzzle together. And at its peak, we had temporary files raising to the level of 750 terabytes worth of data, all on side of the platform to put this puzzle together. So that was a really exciting one, and I was really excited to see that I didn't ruin anybody's Christmas by bringing the cloud down and having my colleagues call me up yelling at me, what was I doing? We've been involved in this project that Angela mentioned earlier, the Vertebrate Genomes Project, where they're aiming to make a de novo genome assembly for all 65,000 different species of vertebrates, which is really cool. But this year, we had our first release just a month ago of the first 15 species of that project. And these are incredibly high resolution genome assemblies. They in many cases exceed even the human assembly that we work on today, in that not only are we attempting to create sort of chromosomal level assemblies, but we're also attempting to make sure that they're phased. So if they're diploid, you have an idea of the what variants go with what variants and what have you on a single chromosome. And then finally, most recently, we've been involved in this really exciting project with Doreen Ware and Kelly Dawes about looking at various maze assemblies. And we've had some really excellent work results there, but I'm not going to spoil the punchline there, because later we're going to hear from Doreen herself tell a little bit about that project. Okay, so that was our quick assembly portion. Next, I wanted to tell you a little bit about visualization. So last year, we were able to tell you a little bit about some of the work that we've been doing in the visualization arena. So we heard about the importance of visualizations from the St. Jude Project, and I think all of us sort of recognize that the importance of putting the raw data that we use or develop on a day-to-day basis in a visual context that allows us to gain better insight into what the data is telling us, and also makes the data accessible to a wider range of folks, right? Folks who aren't going to be just comfortable at a command line interface or running statistical tests and these sort of things. So I can't tell you all the visualization work that we've been doing over the past year, but I've got 2 quick stories I wanted to share. So the first is the development of this cool tool, DOT, by my colleague Maria Nottestad. And so this is a dot plot viewer. And what dot plot viewers are meant to do are to compare 2 related genomes. So in this depiction here, what you see is a depiction comparing the human genome on the x-axis here to the gorilla genome on the y-axis. And at any point where you see similarity between the two, you put a little dot there, right? So what this is saying is from a macroscopic scale, we are all very, very, very similar to gorillas, right? But we knew that. The fascinating thing is that you can start zooming in at different areas and see where the differences start coming into being. The gross sort of structural differences that help explain differences between individuals or between species. So in this case, we zoomed into a short location where you see this weird diagonal bit that goes sort of orthogonal to the other diagonal, right? So what that is indicative of is that there was an inversion at that location. So we match gorilla very well there, but in some sense, for some reason, it flipped and turned it over. So the dot viewer itself is really cool. It's open source. We highly recommend you take a look at it. Next thing I want to show you is a cool project which we'll actually be demonstrating here. So every Friday, the science team has the opportunity to spend half a day pursuing fun projects. And one of the fun projects that we've been exploring is how one can use VR to make compelling genomic visualizations. So I do encourage you, later we'll have a setup over near where we got breakfast this morning, and you can see what genomes look like in 3D. So finally, I'm going to speed through this because I've been given the 5-minute sign. I want to talk to you real quickly on the work that we did with Parliament 2. So Parliament 2 is a project we were directly involved with our friends over at Baylor, and it's tasked with sort of developing ways to detect structural variants in sort of short read sequencing in this case. So that's a challenging problem because many of times the events that you're looking at are larger than the reads that you get off. And there are a lot of different tools available for you to detect these, but none of them are particularly good. And so this is an ensemble approach that takes from all the different short read callers of structural variants and then tries to create a consensus call. So let me show you a couple of key slides there and then wrap up. So shortly, the individual tools themselves are challenging to install, and they're not always optimal to run. You can see in the blue, some of the tools take a long time to run, but after the science team gets hold of them, we're able to do clever things like parallel cross chromosomes and distribute them across different machines and bring that time down dramatically. Then in Parliament itself, so the individual tools rarely get above 50% precision. But when combined together, you can see that you're able to get precision for deletion events up to 100%. So that's exciting. And then for recall, you're able to get from down here at the right-hand side is showing you that individual tools by themselves have very low recall, but when you combine them, you can get up to 71% recall. So again, the idea is that together we're better than apart, right? So the nice thing that we're able to do when we did this study is we can not only improve the precision and recall, but we can get an idea of how accurate the variants themselves are. And so we were able to create a heuristic that gives you a quality score based upon this variant, what tool called it, and how many tools agreed with it. And based upon that, assign quality values to the individual structural variant calls. Okay. So this work is being used at Baylor as part of their CCDG project. And even Tony Blair likes it, who knows a thing or two about Parliament. And it resulted into a paper which we have just recently, the past few weeks, released on bioRxiv with folks at DNAnexus and at Baylor. So if you have any more questions or interested, please go check out our paper. And with that, I think I have a minute to spare, and I'm happy to take a single question probably. But thank you all for your time.